In this module, we again talk about the batch culture kinetics. In previous modules, we talk about a linear regression equation, we would call as the equation which we can utilize for the estimation of mu, which we called as specific growth rate constant. But that equation was only applicable when the cells are unicellular. But as we know that when we have the mass culturing of the organism, and then we can also have the chance to grow the unicellulars, in which we include yeast and bacteria. But we can also utilize the multicellulars, mostly filamentous fungi and different other cell suspension cultures. But mu as concern is only applied when the cells are unicellular. Each and individual cells have the tendency to divide and have the equal chance to interact with equal substrate independently. But in case of multicellular organisms, some cells are growing at one time while others are at rest, same time. So at the one time, different cells are at different phases of their growth. Some are at log phase while others are at stationary phase. That's why we cannot calculate the mu properly in that case. So as we say that dx over dt is equal to mu x, and while dh, the change in height divided by the change in time, can be called as mu h. Sometimes the growth only happen in an area, so we can call as the d change in area, dA over dt. So that is equal to mu a. So here the h is the total length and the a is the number of growing tip or change in the area of the colony. So by using these equations, we can easily calculate the mu. But in submerged fermentation, a mycelial organism, when we talk about the filamentous fungi, that may grow as dispersal of hyphal fragments or as pellets, the growth of pellet will be exponential until the density of the result in the diffusion limitations. So under such limitation, when the central biomass of the pellet will not receive a supply of nutrient, nor will potentially toxic secretions or the product diffused out. So thus the effect of growth of the pellet, only growth proceed from the outer shell of the biomass, which is actively growing zone. So you can see that if any fungus grow in a pellet, so only that the tendency of the mycelia grow at the tip, on the periphery of that. So because that have the most chance of the nutrient supply, but as concern those cells that is present in the center of the colony, they having some effect of their secretions, they have not the equal chance of growth. So growth only happen on the tip of, we can say that the periphery of the colony. So in this case, we cannot properly have the linear growth pattern. We can have the correct specific growth rate constant. So in this case, we have to be another equation derived by the PERT. In this PERT give the solution of this. Here you can see that an equation that is the transform of that the previous equation, in which you can see that ln xt is equal to ln x naught plus mu t. But here you can see that m, that is m naught, is the mycelial mass at time naught, and mt is basically the m mycelial mass at time t. So if we take the under root, cubic root of the mycelia, and we plot against the time, then we have a straight line. So in this case you can see that, so if we have time on x-axis and m over 1 over 3 on y-axis, then we have a straight line. In this, the slope of this, that is the K. So K is also equal to the mu, which we called as specific growth rate constant in term of when we talk about fungal hypha or the mycelia. And we know that the fungal hypha are mostly known as the mycelium. So this equation can be used for the estimation of specific growth rate constant in term of fungi.